welcome to Proving the Point, where I talk about important research papers that you should know about. Today I'm going to discuss this paper from David Reich's lab called Ancient DNA Reveals Pervasive Directional Selection Across West Eurasia. And what they did in this paper, as the title suggests, is they basically took a bunch of ancient DNA samples of Europeans and analyzed the mutations present in those genomes to see which genetic changes were happening in ancient Europeans and how that impacted the traits of Europeans across time. So they looked over a 10,000 year time window in this paper. So the reason this paper matters, before we get into the methods and the results, is that a lot of people today, they argue that there are no meaningful genetic differences between the races or the human populations in general. They like to argue that humans are all just born as a blank slate and that the only difference between different people groups has to do with environmental factors and social conditions. But this and many other papers show that that's not the case and that there are a lot more genetic differences that impact important biological processes. So we'll talk when we get to the results about what types of processes are affected, but it's things like metabolism, immune function, and cognition and intelligence. So we're going to talk about that as we go. So we'll talk more about why it matters toward the end, but first we're going to cover a little bit of the methods of this paper and then, like I said, focus mostly on results because that's what's important. But for the background of the methods, like I said, they basically took a bunch of ancient European genomes, around 16,000 ancient European genomes, and analyzed their mutations present in those genomes to see which genetic changes were happening in Europe and how those changed across time. So certain genetic changes became more common in the European population, others became less common. That's positive selection or negative selection, respectively. And that seriously impacted the traits of Europeans. So, like I said, we're going to get to the results later, but just as a preview, among the things that were affected were genes related to metabolism and fat storage, genes related to psychiatric conditions like schizophrenia and bipolar disorder, and importantly, genes related to cognition. Specifically, a lot of mutations were occurring in Europeans that are associated with increased cognitive performance. All right, so that's mostly all we really need to say about the methods. It gets a lot more technical and detailed than we need to get into, but I guess just to highlight some unique features about this paper. So other papers have done similar studies before, but with this paper, they added around 10,000 new ancient genomes. So that massively increased their ability to draw conclusions, and they combined that with previously established genomes for a total of around 16,000, as it says, spanning 18,000 years. And then they also used a few thousand modern people, so that way they had something to compare it to. So again, these were people with European descent. Ultimately, they found around 8 million unique mutations or gene variants in these populations, and many insertions and deletions, which basically just means a nucleotide in the genome is added or deleted. So they focused on chromosomes 1 through 22, so those are the autosomal chromosomes, meaning not the sex chromosomes. Again, the details of what they did are a little bit more technical than we need to go into, but basically you can estimate what's called the selection coefficient for a mutation, for an allele, a gene variant, to see whether or not that gene variant is being positively selected, it's selected for, or whether it's under negative selection, being selected against. And geneticists do that based on the fitness associated with an allele. So if an allele has a fitness advantage, then it has a high selection coefficient. If it has a fitness disadvantage, it has a negative selection coefficient. Okay, the way they do it with ancient genome samples is they basically look at how a particular allele, a gene variant, changes across time. So if, for example, they analyze one set of genomes from 5,000 years ago and another set of genomes from 2,000 years ago, and they see that a particular gene variant has become more common, then that means it was probably under positive selection. It was being selected for, because the people who had it had higher fecundity and reproductive success. All right. So like I said, they go into a lot of detail with how they evaluated their method and confirmed that it's working properly. So they tested it against like real data. They tested for potential confounding factors, other forces that could have been influencing changes in allele frequency besides like positive selection. But again, we're not going to go into all that. What they ultimately found, so getting into the results, is that there were around 479 loci, which basically just means like genetic areas in the genome, so like specific genes mostly, that reached their like significance threshold for being under directional selection, positive or negative. Okay? In other words, there are around 479 unique genetic locations in the genome that were under some form of directional selection, positive or negative, in these Europeans across this 18,000 year span. Okay, and we're going to look at what some of these genes were and what their effects might be on the body. Okay, so we're going to skip some of these early figures. A lot of this is just them proving that their model is working properly and that their methods are sound. But again, it's more technical than it's worth going into for us. The first important figure that we're going to look at is figure 2a. So what this figure is showing is on the x-axis, it's each of the 22 autosomes, the 22 chromosomes besides the sex chromosome. And then on the y-axis, it's showing basically how much a given gene was under directional selection, which again, that means either it's being selected for or it's being selected against strongly. Okay, and the way that you interpret this graph, it's called a Manhattan plot, is basically each dot represents a unique genetic allele at that specific position on the chromosome. So for chromosome 1, it's got, thousands of genes on it, and at each potential position, there are different genes that can be mutated, therefore giving rise to a different version of that gene. So, for example, this IL-1 gene, that's interleukin-1, is a gene involved in immune function. This dot indicates that there's a variant, a mutated variant of IL-1, and the fact that this dot is high up on the y-axis means that it's under some form of directional selection in these samples that were analyzed, meaning it was either becoming more common across time, meaning it was beneficial to the fitness of the individuals who had it, or it was becoming much less common across time, meaning it was deleterious to the people who had it. Okay, so each of these different abbreviations is a different gene. So CCR5, for example, is also involved in immune function. There's various other genes involved in, for example, this one, major histocompatibility complex, that's also involved in immune function. So we're going to see on the next couple figures what some of these genes do and why they're important. But this is just showing there was substantial, meaningful selection on all these different genes acting across that 18,000 year time span. And this line, this black line that crosses it, that's their statistical significance threshold. So if a gene variant, an allele, is above that line, that means it's under significant selection. And again, the higher up, the more significant that selection is. So again, these two panels are not necessarily relevant to the main point that we're trying to get to. The big figure is figure 3, where each of these panels is basically a different gene version, like a different allele for a given gene. So like right here, there's the ABO gene, that's what controls blood type. Okay, this part after the gene name is referring to the variant that is the mutated variant present in this sample. So in other words, what they found is that there was this blood gene mutated variant right here, and then this is showing down here the change in this variant across time in these European people. So it used to be the case that this gene was very uncommon, rather this gene version was very uncommon. And then across time, there was positive selection for this gene variant, making it more common in the population over time. Okay, so the y-axis is basically prevalence of this variant of the gene in the European population at different points in time. So again, 8,000 years ago, it was very uncommon to have this particular version of this gene. Across time, it became much more common in the population. Okay, so what that means in terms of your biology is this is showing blood type B. So in other words, this gene version makes an individual more likely to have type B blood. Okay, or look at this one. There's this gene, TCHH. Apparently it's involved in hair growth, and this particular gene variant, this mutation in this gene, is associated with increased hair straightness and increased baldness. Okay, but over time, the prevalence of this variant is actually decreasing in Europeans. In other words, like the gene variant that causes baldness is becoming less common across time in this sample. Okay, so we could go down the list. Here, for example, is a gene variant involved in tuberculosis susceptibility. So initially, this variant that causes tuberculosis susceptibility is very, very low in the European population, but across time, it becomes much more common. And then, my guess is there was a tuberculosis outbreak somewhere around here. It underwent strong negative selection. In other words, this variant of this gene that causes increased susceptibility to tuberculosis started being strongly selected against. Okay, and again, you don't really need to pay too much attention to these numbers. The S is the selection coefficient, which is basically, again, if it's positive, that means it's under positive selection. If it's negative, it's being selected against. This chi value is basically essentially the magnitude of the change and its significance also. So initially it's under strong positive selection, and it enters very strong negative selection. Okay, this gene version is involved in multiple sclerosis susceptibility. So again, it starts off very uncommon in Europeans, but it becomes more common across time until you get to around 2,000 years ago, at which point there's negative selection. Okay, this gene variant involved in hemochromatosis, which is basically like excessive iron intake by the body. That becomes more common and then starts to decline. This gene variant involved in HIV resistance, it becomes more common in Europeans across time. These ones are all more what you might expect to have seen, but these genes are all involved in light skin and light hair, and they become more common in Europeans across time. Okay, all these different genes impact that process. Okay, some of these, this is like showing one that doesn't really change that much. Okay, so genes involved in susceptibility to celiac disease, which is an autoimmune disease of the intestine. That becomes more common over time. Arthritis susceptibility decreases over time. Ferritin level, that's involved in storing iron, increases over time. Crohn's disease, another autoimmune disease of the intestine, increases over time. Leprosy resistance increases. Blood pressure, The variant that decreases blood pressure increases. So in other words, Europeans experienced a mutation that would decrease their blood pressure. Maybe we'll talk more about that in a future video about blood pressure differences between different human populations. Okay, vitamin D levels, the gene that helps control that, it had a variant that increased because, of course, in Europe, there's less sunlight, which means they need genetic changes that increase their ability to acquire vitamin D, which is produced with the help of sunlight. This gene variant lowers the risk of alcoholism, so this became more common in Europeans, meaning lower risk of alcoholism for Europeans. Okay, gout, which is a type of arthritis. Hyperlipidemia increased, that means too much lipids in the blood. Okay, and then there's a variety of others. So those are some of the genetic changes that were happening in European people across this roughly 10,000 year span. Now what's really interesting though is their next figure, which talks about polygenic traits. So these traits up here in figure 3, they're mostly related to single genes. So in other words, like, let's go back to the blood type example. The ABO gene is a single gene that controls blood type. Okay, but not all traits are as simple as one gene controls blood type. So for example, with intelligence, it's not like there's the intelligence gene. Rather, there are, hundreds of genes that influence intelligence. The human brain expresses most of the genome. Most of the genes of the genome are functioning in the human brain. So traits like that are called polygenic, meaning multiple genes control that trait. Okay, so they had to do a slightly different analysis, but the same general way of interpreting this graph as before, which is basically the x-axis is time, and then the y-axis is prevalence of that particular trait. So like here we have darker skin tone. We already saw in the previous figure that many skin tone genes were, the alleles that control lighter skin tone were becoming more common, while genes that control darker skin tone, those alleles were becoming less common over time. Again, that's all related to sunlight availability and vitamin D necessity. Here, body fat percentage. So genes involved in body fat percentage were also becoming less prevalent. I should say alleles controlling that were becoming less prevalent. So Europeans have lower body fat across time, similar to waist circumference and waist to hip ratio, which are both signs of metabolic and general health. The alleles that controlled those were declining across time, signifying an improvement in health in Europeans. Alleles that produced bipolar disorder, those alleles became less common over time. Alleles that produced schizophrenia were becoming less common over time. Alleles involved in, this is basically just saying alleles involved in like addictive behavior become less common over time. So basically all of these are showing improvements in the genetics related to neuropsychiatric health in Europeans. Okay. Overall health decline. So these are gene alleles involved in poor health. They're becoming less common in Europeans across time, signifying an improvement in overall health. Okay. Alleles involved in walking pace. So if you don't know the speed at which people walk, that's positively related to intelligence and it's positively related to overall health. So this is basically another indicator of overall health and intelligence. Anyways, alleles controlling walking pace were increasing over time in Europeans, meaning basically their walking pace increased over time due to genetic changes. Alleles involved in intelligence increased over time, meaning Europeans underwent mutations and changes in mutation frequency that altered their genome in a way that made them have higher intelligence. Genes involved in household income, alleles that benefit household income increased over time. Again, that's basically just a placeholder for intelligence or like work ethic and motivation. Same with years of schooling. So a lot of these are indicators of intelligence or forethought and planning and things like that, increasing over time. So what these show is an increase in basically overall cognitive function due to genetic changes happening in Europeans over the past 10,000 years. Okay, so that's the main takeaway from this paper. They have more data in their supplementary information at the bottom, but most of it's not super relevant to the point we're trying to get across. One interesting figure from the supplement is just this one right here. It's basically a correlation matrix showing in their analysis which traits correlated to which other traits. Okay, so like if you look at body fat percentage, okay, waist to hip ratio is positively correlated with that. So if you have a high waist to hip ratio, that means there's you also likely have high body fat percentage. Okay, like I said, with walking pace tends to be negatively correlated with poor health metrics, such as, again, high waist to hip ratio. So, faster walkers tend to have a lower waist to hip ratio. They have a lower body fat percentage, lower waist circumference. That's any box that's red, that means there's a negative correlation between the two factors. Okay, but things like years of schooling, those are related to walking pace, okay, positively. They're negatively related to poor health metrics like smoking and things like that. So if you want to pause and take time to look at this, you can, just to see which traits are related to each other. Okay, but that's basically the paper in a nutshell. Again, why does this matter? Well, it matters because Europeans have a very distinct genetic history, and that genetic history is largely being erased due to mass immigration and low fertility rates among European people. So if we're going to fight that, we need to continue to argue that Europeans are genetically distinct and that they actually have certain positive genetic traits that should be preserved. So, thank you.